So why does the cell cycle progress? Is it simply like a row of dominoes? When you knock the first one over, the rest of them fall? Or are there specific signals that transition the cell from G1 to S to G2? This is a question that researchers had. Now, cancer cells escape this regular control of the cell cycle. So what is it that controls the transition from G1 to S to G2 to the mitotic phase? Researchers set up an experiment to test what controls the cell cycle. So there are certain drugs you can apply to cells in tissue culture that will arrest them at different stages of the cell cycle. So if you have a plate of cells growing in tissue culture, you apply this drug and the cells will continue to go through the cycle till they get to a particular stage. So in this experiment, they locked cells in S phase of the cell cycle, and then they had other cells that were in G1 of the cell cycle. So they knew what stages of the cell cycle they were in. And then they fused these cells together, forming a hybridoma. When you do this, what happened was the cells that were in G1 jumped into S phase, so they immediately started replicating their DNA. Now, from that alone, that doesn't really answer our question, because after G1 comes S, so it looks like they just jumped into the next stage of the cell cycle. So the researchers then asked, is it possible to skip a stage in the cell cycle? So this time they started with cells that were already in mitosis and cells that were in G1 of the cell cycle. They have not yet replicated their DNA. And when they fused these cells together, the cells that were in G1 that had not yet replicated their DNA began to go through mitosis. They began to go through prophase. The nuclear envelope began to break down. The DNA started to condense. The mitotic spindle started to form. And so what this suggested was that there was some positive signal here from the cells that were in M phase that caused the cells that were in G1 to start going through the steps of mitosis, even though they hadn't gone through S phase yet. Many more experiments were involved in determining that the stages of the cell cycle are regulated by checkpoints that prevent the cell from going into the next stage. And then when those positive signals are present, it allows the cells to skip from one stage of the cell cycle to the next. So there's a hard stop at the end of G1 called the G1 checkpoint that checks to make sure that there are sufficient nutrients, that the cell is stable, and that it should proceed into S phase and do DNA replication. Once you get through S, G2 pretty much happens automatically, but then there's another checkpoint that says, hey, do we have enough food? Do we have enough resources? And also, was all of our DNA actually replicated before the cells move from G2 into mitosis? And then before finishing mitosis, there's another checkpoint, the M phase checkpoint, that says, hey, did we actually finish mitosis? Did we separate the chromosomes appropriately before the cell finishes dividing and we move into two new cells that do G1? Some cells basically constantly cycle as long as they get the positive signals that say you can proceed through one checkpoint or the next. The cell cycle will continue. So this is true for our skin cells or our digestive epithelium. But other cells will get to this G1 checkpoint and they'll just stop. Okay? And in fact, these cells will stop cycling altogether. They'll exit the cell cycle and go into a stage called G0. These cells are non-dividing or senescent or quiescent. They don't divide anymore. So this would be true of most of our neurons, for example, which have exited the cell cycle and are just going to do their job as a neuron and not divide anymore. Those positive signals that regulate those checkpoints are made up of pairs of proteins called cyclins and cdks, or cyclin-dependent kinases. The cyclins get their name because they cycle. In other words, their concentrations fluctuate with the stage of the cell cycle. but they only work in the presence of their partners, the cyclin-dependent kinases. So their name describes what they do. They're cyclin-dependent, meaning they only work when their partners, the cyclins, are present, and they're kinases. So what's a kinase? Kinases are enzymes that add phosphorylation. And then the opposite enzymes are called phosphatases. Phosphatases remove phosphorylation. And the presence or absence of phosphorylation acts as a switch that turns proteins on and off. Now, phosphorylation doesn't necessarily mean the protein's on, and dephosphorylation doesn't necessarily mean the protein's off. For some proteins, phosphorylation turns a protein on and dephosphorylation turns it off. But for different proteins, phosphorylation turns a protein off and dephosphorylation turns a protein on. The thing that's constant is kinases add phosphorylation and phosphatases remove phosphorylation. And so CDKs are cyclin-dependent kinases. They're enzymes that phosphorylate target proteins when their partners, the cyclins, are present. So the way that these cyclin CDK partners were discovered came from these studies where they had cells synchronized in different stages of the cell cycle. So in that initial experiment that we talked about, they took those cells that were in M phase, and there was something in these cells, some factor that was present that caused these cells in G1 to jump into the mitotic phase. And so the scientists wanted to figure out what that thing was. So they purified the cytoplasm from those cells that were in the mitotic phase. And they saw that that cytoplasm, if they added it to cells in other stages of the cell cycle, caused them to jump into the mitotic phase. And so they split that cytoplasm mixture that was full of proteins and nucleic acids and carbohydrates into smaller and smaller fractions. And what they saw was there was one particular subset of the cytoplasm that was able to cause other cells to jump into the mitotic phase. So they called this fraction the MPF activity, or the maturation promoting factor activity, or the mitosis promoting factor activity. And then they further isolated the components of that fraction and saw that there was one important protein that correlated with this spike in activity, and that was the cyclin CDK. But the CDK by itself didn't do anything. There was another protein that was present all throughout the cell cycle that worked with the CDK to promote progression through the checkpoint. So what they saw was there was this one particular protein, which they named the cyclin, that's concentration peaked at the same time as the MPF activity. But the cyclin protein by itself could not lead to progression through the cell cycle. The other protein that was necessary to get the MPF activity was the cyclin-dependent kinase. The concentration of the cyclin-dependent kinase was constant all throughout the cell cycle. It's always there. Okay? But the cyclin concentration starts going up around G2, and as it peaks in the mitotic phase, it promotes progression into the mitotic part of the cell cycle. So it's these partner pairs, the cyclin cycle. The cyclin's concentration goes up and down throughout the cell cycle, but the cyclins don't work on their own. They need their partners, the cyclin-dependent kinase, which are always present together to promote progression through the cell cycle. And there are different cyclin CDK pairs that regulate the different checkpoints throughout the cell cycle. So the cyclin CDK pair that regulates the mitotic phase checkpoint, that kinase activity phosphorylates proteins that lead to chromosome condensation, nuclear envelope breakdown, and growth of the mitotic spindle to promote progression through the mitotic phase of the cell cycle. And then the last thing that the cyclin CDK pair accomplishes is it activates the enzyme that breaks down the cyclin. So this drop in cyclin activity is actually a negative feedback loop. Cyclin CDK pair turns on everything for mitosis and then shuts itself off so that the rest of the cell cycle can proceed. So there are different cyclin CDK pairs that regulate each of the checkpoints through the cell cycle. This picture shows just a single CDK interacting with different cyclins at different checkpoints, but really there's a unique cyclin-dependent kinase that matches with a unique cyclin for each cell cycle checkpoint. But what they have in common is that the cyclin-dependent kinases are always present at a constant level throughout the cell cycle, whereas the cyclins fluctuate in concentration throughout the cell cycle and thus regulate the transition through the different cell cycle checkpoints.